Okay, so welcome everybody. Welcome to today's seminar. We have today our first PhD talk of the 2025. We have here Manuel Serrano, who is going to present his PhD project. He's a biologist. He did the master in biology and conservation biology at the University of Pablo de Olavide, and he has done some his undergraduate and his master thesis also in Doñana. And now he's working at the EBD, at the isoplastic group, and he's working with aquatic birds and how they disperse plastic. So welcome, Manuel, and the floor is yours.
Thank you all. I'm Manuel Serrano, as Isa told, and I'll be presenting my thesis plan, talking about the aquatic birds as dispersal vectors and the plastic ecotoxicological implications and the role of the trophic ecology and movement. I've been working in this thesis for now 10 months, so there's still a lot to do, but I'll show you what we've been doing in those months. Well, the first thing that we notice is when looking at any searching web like Web of Science, we notice that most of the papers that have been published are talking about those plastic and the contamination, but related with the sea and the ocean and everything. But when looking at more inland and freshwater ecosystem, marshes and so on, we see that there are few of them, there are less papers that talk about this, and all of them also with the marine relative only in the last few years. When talking about the plastic, in this case birds related to those ecosystems, we see that multiple studies show how those plastics are having direct physical impacts on the birds, and talking about amputation, stranglements, and all these diseases that are damaging the birds. But not so many are talking about how those birds are taking those plastics that are damaging them and moving into natural spaces, and also even few talking about how those plastics that are also correlated with contaminants and other pollutants are maybe damaging also the health of the animals. So there are gaps of knowledge that we want to fill, and are basically those, the importance of the aquatic birds and as biovectors of those plastics that they are taking from urban areas and landfills, for example, and they are taking it to natural spaces, and how those toxics are affecting the health of the animals. So in the framework of the thesis that is inside the Isoplastic project, we are aiming to analyze the plastic loading via also the movement connectivity data set, and a characterization of the items that we are finding inside those pellets, inside the nest they are building in the colonies, and now moving to the toxic compound, how those toxics are hitting the birds and making the health of the birds worse, and also via stable isotopes analyze how the trophic level of those birds and the foraging strategies are changing. So the first three chapters will be talking first about those movement and information that we have, and how are those birds dispersing the plastics, and then the last two will be talking about the ecotoxicological implication of those birds being close and in touch with those plastics. We've been talking about the aquatic birds, but our models will be the following ones, that are two gulls, the yellow-legged gull and the lesser black-backed gull, the cattle egret and the white stork. The importance of choosing those animals is that they all are easy to find in urban areas and landfills. They are foraging in those places, but they are also related with natural areas as marshes or wetlands. So they are doing daily movements from those areas where they are sleeping, or maybe they have the breeding points, to those anthropogenic areas where they are foraging and taking the food, or they are foraging. So they are connected both places, one that should be pristine and the other one that is usually found with plastics. So the colonies and the sleeping point that we have for the species are those ones, and we selected the most important ones for each of these species, but all along Huelva, Seville and Cádiz. But not only that, also we took samples from Chafarinas for the yellow-legged gull, and all along Morocco for Bubulcus ibis, the cattle egret, thanks to Maria that went there a couple of weeks ago. We've been talking also about sampling those points, but what kind of samples we've been taking and working with. Mainly pellets of all of the species, but also nests, eggs and other biological tissues to analyze the contaminants. And also we are working with GPS tracking devices, so we know the movement of the animals. It is a multidisciplinary approach of the problem, and using movement ecology, toxicology, histology, statistical modelization to estimate how many plastics and how much plastics they are bringing to natural spaces, and trophic ecology to know how those kind of ecosystem are changing the way of those birds to forage. Also analytical methods via F-tier to analyze the plastics, and also image analysis, stable isotopes and GPS tracking data, and ecotoxicological analysis, for the biological samples. What samples do we got till now? Well, there is still one column left that is gonna be the sample that we still need to do in this breeding season in 2025. And we are taking those pellets, but also we have 50 nests from both of those species, and the yellow-legged gull and the white stork, 10 eggs from yellow-legged gull, and also from the yellow-legged gull. We've been doing 45 necropsia at Gibraltar, where we took a lot of biological samples that we'll be talking about in a minute. And jumping now to the first chapter, that is settled in alluvial marshes, we'll be aiming to quantify how many plastics they are bringing, the two of the species of gull that we are working with, and try to find difference between the two species of gull that they are foraging in those places. And our hypothesis is that yes, both of the species, because of their foraging strategy, will be bringing quantity and types of plastics to the alluvial marshes, and the quantity will be huge. So yeah, we've selected those two species that are the ones with, we think, more impact on those plastic vectoring, that are the yellow-legged gull, that is more coastline related with talking about foraging and with a more natural way of foraging, and the laser black-backed gull, that is a more inland and foraging style bird. Of both, we have more than 200 pellets from 2022 to 2024, and as like the GPS data that we have from those two species. And we are gonna do F-tier analysis and image analysis of the plastic item that we found inside those pellets. So the first thing we needed to do was going to fieldwork and grab the pellets, and then moving into the lab, we via stereo microscope we selected the items, and then we sent them to the University of Cádiz to do the F-tier analysis over there. We also do a physical analysis of the items found, and that was the color analysis, but also a size categorization in micro, meso and macro plastics, and a morphological characterization in hard line, foam and film. This is important, this is characterization, because it's gonna give us also a lot of information. For example, for the categorization of size, we can know how those plastics, depending on the size, are being dispersed more or less, and also the origin for example, with the morphological characterization, because maybe the lines are coming from sea work and everything related to the sea. And at last, in the methods, we'll be talking about also a little bit about the deposition model. That is component that is gonna give us the daily plastic loading to the marshes, and it's related with the number of individuals of both of the species that are daily in the marshes, also with the gestion rate of pellets that we settle in 1, because we wanna keep conservative in this study, even though we know they can drop more than 1 pellet per day, and we wanna keep this in 1. And also a random plastic pellet plastic weight from all the pool of plastics and pellets that we have is going to be included in the model. So for the first results that we got for this chapter, we see that the laser blackback gull is the one that is foraging, and we found plastics more usually, with only like around a 27% of them free of plastic or any anthropogenical item. And the yellow-legged gull showing less, but well, less, because the 60% of them were showing plastics inside. From the FT result analysis, we see that the polyethylene, 2 kinds of polyethylenes are the ones that are more usually found. Those are plastic that are only one use per plastics, and also polypropylene and polyethylene that are 2 plastics that during their fabrication are freeing a lot of contaminants to the atmosphere, and there are also one-use plastics. And now, relating the plastic type with the size of those items, we can see that the microplastics and the smaller mesoplastics are the ones that are more usually found inside the pellet. So those are the ones that are easier being dispersed by the gulls, and also the hard ones, because those are the ones that are breaking easier. In terms of where those gulls are dropping the pellets, we see that they are not randomly dropping the pellets in the ODL marshes. Most of the gulls are usually in those places. This is the Liebres Island, is where the colony of the yellow-legged gull. Those analyses are only for the yellow-legged gull. I will do the same for the laser blackback gull once I got the tracking data. And the breeding colony is settled over here, so it's more likely to us to find those pellets here, and because of that, all the plastic that they are dropping. But not only those, this place also just in front of Punta Umbria, that is the urban area where they usually are foraging more. This is the daily use of habitat, so those are the hours of the day, and those are in different colors the places where they are foraging. We see that at night they are usually sleeping or staying in the marshes, and during the day they are going away to crops or ocean, but more important to urban areas and landfills, where they are usually, or where we suppose that they are foraging and taking those plastics. And we also saw that there is during the year there is a differentiation between the places they are foraging in. Because during summer and the breeding season, more gulls are going to urban areas. Maybe because a lot of people is going there for vacation, so more debris are being generated, and gulls prefer going there that are only a few kilometers away from the marshes than going to landfills that we can see here that in those months they are visiting less the landfills, because those are 15 and 30 kilometers away from the marshes. So if they have more places to forage or more food next to the marshes, they prefer going over there. For the analysis of and estimations of the plastics found, we see that for the yellow-legged gull, 72 kilograms of plastics per year are being brought only by pellets, not analyzing the nest yet. And for the laser blackback gull, over 191 kilograms, and if we put them together, more than 250 kilograms of plastics per year, and I said only with those 2 species, and only by pellet dropping. Now we knew what the impacts on the marshes and with these species, and we wanted to change the species and to change the ecosystem. So we went inland to a freshwater lagoon with the storks, and we wanted to know how many plastics they were bringing to those kind of ecosystem, knowing that this species is having a different foraging strategy, but going also to landfills and doing those kind of movements. And also determine if there is any pattern of preference on taking different kind of plastics. Our hypothesis is that they will be bringing a lot of plastics too, to, in this case, the ESA de abajo, and they will show a pattern in, because they will be preferring taking some kind of plastics to build a nest, for example, than others. Till now we got 20 nest sample in 2024, and we are aiming to take more plastics from the surface of the nest in the ESA de abajo, and more than 100 pellets in the same years, 2023 and 2024. We still need to do the pellet and the plastics in the nest analysis, and you might be thinking why, if the ESA de abajo is close to Doñana, it's a place that is is protected and everything, that is here, there is the lagoon, and Doñana will be over here and over here, but there is Paco. Paco is I don't know. She is a stork that is breeding in the ESA de abajo, and she's doing daily movements to Santa Marta's landfill. So even though this is a protected area far away from urban areas and everything, though this is the importance of studying this kind of animals, because they are connecting places that should be keeping pristine as the ESA de abajo and protected areas with landfills when they take all those plastics and so on. So after sampling the nest, we found that there were a lot of plastic in there, and they were using them a lot to build like stronger nests. So in the following chapter will be analyzing those plastics, because we were lucky to have a certain sampling that I will talk about in a second. So we want to see if there is difference between the nests that are farther away from the landfills and the ones that are closer, and see if the ones that have less distance to landfills show up with more plastics. So and our hypothesis is that yes, they will show up with more plastic, the ones that are closer to landfills. We were lucky because there is a line of power towers that cross from Huelva to some villages next to Seville, and we were able to take samples from all along the line, and just in the middle there is 2 landfills, the one in Niebla and the one in Villarrasa. So we kind of have a transect, and that is gonna show us if there is any interaction between the quantity of plastics in the nests and the distance to the landfills. And those are the towers. As you can see, there are a lot of nests, and the importance also of those kind of samples is that they are dropping those nests every year. So every year they are building them again. So the plastics inside those nests are only are being analyzed in a single year. I mean, and those plastics are being brought to the natural spaces and to the towers year by year. No, they are not 3, 4, 5 years old plastics. So we can estimate better how many plastics they are bringing per nest to natural spaces. And thanks to SEMI, we've been able to, and the technicians, we have been able to drop all those nests and analyzing them in the same place, and taking all those plastics to here EBD and then analyzing them. Now that we've been talking about how those birds are moving all those plastics, we wanted to know how those plastics are affecting directly the birds' health by studying the pollutant that they have, with they I mean the plastic they have related, and how swallowing and moving with all those plastics are affecting the health. So with gulls in Gibraltar, we want to analyze the presence of plastic derivated pollutant inside the different tissues and sample that we've been able to have to take, but also the damage of those plastics are causing to the digestive tract of the birds. And also we got movement data that we can relate somehow on how those birds are foraging in this certain colony. And our hypothesis would be that the contaminants related to the plastic will show up in the samples, and we expect to find some damage inside the tracts of the gulls. I think quite important this colony, because it's a place where we have every single sample that we've been talking about. We've been doing necropsies that I will tell you now, and the samples that we've been taking. We have pellets, we have nets in the following breeding season, GPS tracking data to confirm the results that we'll be having here. And for the necropsia we have been taking muscle, liver, intestine fat, lung, kidneys and brain, and also fat from the European gland for the toxic analysis, feeder for stable isotopes analysis, and also morphological measurement, because we have also the birds over there. We also took the whole tract of the gulls for two reasons. First, to analyze the plastics inside those tracts, because we are always working with pellets, but knowing the plastics inside the tract and the issue that they are showing is quite important. And also we are collaborating with University of Seville, because they are making us some cuts, histological cuts of different parts of the tract to know if there is any issue in the whole tract. And finally, in the fifth chapter, we want to know if there is difference between the colonies and species and of the different animals, and determine if there is any variation depending of the pellets they are found in with plastics, and know if those if more plastic in the pellets is also related with more issue inside the animals and less health patterns. So we got 3 of the animals marked. We've been marking personally gulls and cattle egret, and I'll be talking a little bit more about the cattle egret, because it's the one that we've been having a little bit more of struggles. We've been marking them with Ornitella GPS tracking devices, and we've been tracking them in Costa Ballena, village next to Cádiz, and in La Cañada de los Pájaros, next to Doñana. And they are now currently foraging in crops and heather. We are also planning to mark them in Málaga and in other points of Cádiz to compare the different colonies and movements. For all the species, we've been always taking samples to analyze biochemical analysis, telomeres, length studies, and doing stable isotopes analysis to determine how those birds are how the physical condition and health condition of these birds. We've been working 10 months, and this is more or less how those chapters are keep going. And I have to say that isoplastic is not only us in EBD. There are a lot of people involved in the project and in my thesis, and I have to thank them all for helping me and everything. Also to Lea and Lea Labs here in EBD, where we are analyzing most of the sample, and a special thank to 2 of my bosses, Manuel and Marta, for helping me in those 10 months and letting me realize this investigation. So thank you all for the attention, and feel free to ask any question if you have it.
Thank you, Manuel, for this wonderful talk. I'm sure that there will be questions that could give him some feedback for his thesis. Yeah.
So you're working on different species and they have some different characters, different sizes, and they focus their life in different areas. Are you doing some direct comparisons, or do you have do you have an expectation of differences because of those biological features of them?
For example, the one where we are finding less plastics are the smallest one. I'll move to the, for example, the cattle egret and the gulls. Because mostly, for example, for the storks, and I didn't put any picture, but they are moving a lot of plastic, for example, for the nests. Per nest, they are moving kilos and kilos of plastics in those nests. And comparing, for example, with the nests of the yellow-leg gull, they are also with a lot of plastics, but it's not as impressive as the storks one. And the same with the pellets. The pellets of the storks are this size, more or less, so they are usually bringing more plastics than the ones, for example, from the cattle egret that are also with a lot of plastics, relatively, but not the same quantity as the storks one, for example. So it's something to compare once we do the model, of course.
And the distribution of the kinds of plastic, it's the same for the different species?
This is something that we want to know, and what kind of plastic they are preferring to take and everything. For example, we found a lot of like big pieces of plastics and clothes in the stork nest, because it's quite easy for them to bring them to the nests, and will make a better nest for them, because it will be a better structure.
But in the ones that are digested?
And we don't know yet. But for example, they are all tending to take, for example, rubber bands, because they get confused and they take it as food, or maybe because they think they are worms or whatever. And other plastic that are related with the rest of human consumption, for example, the ropes from meat and everything, they are in all the species, for example.
How do you select your sample points? For example, the nests of the white stork. Do you select them randomly, or?
No. We were aiming to take the samples in places where we knew that there were an important amount of the same birds. If we were with a car or whatever and we see 2-3 storks, then we weren't stopping for take the pellets. But for example, in Dehesa de Abajo, we know that there is this huge breeding colony in Brazo del Este that is also close to Seville. There are huge groups of stork that are foraging over there. So in the breaches of the rice fields, a lot of them are stopping, and we can select and take the pellets easier. So the important thing is taking pellets from a place where there are a lot of individuals of the same species and with monospecific groups. Because if we see that in a single group there are more than one species, we cannot differ easily the pellets between the two species, and we prefer not to take them to avoid those kind of noise in the results.
I mean, are all of them close to wetlands, for example?
No, we got ones that are close to wetlands and other ones that are close to landfills, so we can compare between those sleeping point or breeding points that are closer to landfills and the ones that should be cleaner because they are in protected areas or with most distance to a landfill or an urban area.
We have one from Carles online. Thank you for the presentation. Do you have an estimate of the proportion of the food used by these birds derived from landfills? Closing landfills would have an would closing landfills have an effect on the populations?
For sure, closing the landfill will have an effect of the population, but it's something that we don't really think is something that is going to happen. I mean, most of the landfill that we are talking about are open landfill. Now, this is something interesting also in taking samples from Morocco, because in Morocco, most of the landfills are not controlled and are quite big. So, yeah, most of the catalysts are foraging over there, and there is no control in those landfills. So, yeah, all these species are foraging in those landfills. So, closing them will have an effect. But I mean, I don't really think it's something possible at all. We can show the impacts, but nothing more.
Cool.
Yeah, so great talk. I have a question about the plastic composition in the pellets.
Yep.
So at some point you're talking about if they show preference for a specific type. So I'm just curious if it's, how are you going to test if it's a preference, or if it's just a representation of the plastic types that are available? So for example, in the right pie chart, there's these two types of plastics that are found more common, I guess, in the pellets. But I would suspect they're also found more common in the area that they forage.
That's the point of for example, in the last chapter, because in this one we are also only analyzing one place that is close to two of the landfills. But we want to analyze, that's the point of analyzing different colonies of different species in different places that are closest to one or other landfills. So it's something that is quite tricky to know, and we will attempt to know how to defer this. But yeah, of course, if those plastics, for example, are the ones that are being more produced, is something that we need to keep in mind. And if comparing the different places, the different colonies of different landfills, and they are showing up with comparing with model, we see something. It's something that we need to keep in mind, basically.
So would it be something to take like control samples from the landfills just to see the plastic composition that is like natural in the landfills?
Yeah, we've been also trying to catch pellets from the same landfills, but the landfills don't really communicate with us, because it's something that we wanted to know, because the plastic that are moving to those landfills by the trucks and the urban consumption are the ones who are gonna be there, and those birds are gonna be eating. So if we knew those plastic composition, would be awesome.
Cool. Thanks.
Thank you for the talk. Very interesting project. In relation to the when you plan to assess the toxicity levels or levels of contaminants in different species, maybe I have missed the point, but do you have how do you set control individuals? I mean, do you have any control, for instance, populations or areas where the birds are not eating on plastic, are not feeding on plastic contaminants? And my other question is a quick one related to the type of toxic materials or contaminants that you find in the birds. Do you know if any of those accumulate in plumage or soft parts of the birds, so that could it be possible to assess the levels of contaminants in museum specimens, in all specimens that we have here in the collection or in other museum, and then compare the levels of toxins that you find in the populations now with past populations, with historic samples, so to speak?
For the first question, that is quite good one. Yeah, it's something that we've been also struggling a little bit about the controls. Which are the controls? Because all those colonies are always foraging in and showing up with plastics. So for the controls, we are gonna try to get the cleanest colonies sampled with blood, for example, and compare them with the same species that are closer or foraging more in landfills and or urban areas, and compare the level of contaminants or via also isotopes, how they are foraging and where they basically where they are eating or not, and comparing the ones that are cleaner with the ones that are closer and showing up with more plastics. I hope I have answered your question.
La idea era comparar, por ejemplo, con sitios menos contaminados como la isla de Tarifa, donde sí hemos estado colectando huevos, no tanto a nivel de individuos muertos, sino huevos, por ejemplo, ahí sí que tenemos. Entonces ahí sí que tenemos varias zonas, unas más conectadas con zonas de vertederos y otras menos conectadas con, o sea, con tipo de uso más marino, de recursos marinos. Y ahí sí que podemos tener eso, pues una amplitud mayor de sitios de muestreo con más y menos influencia antropogénica, ¿vale? En cuanto a lo que son los contaminantes de huevos, que lo que estamos midiendo son plastificantes, sobre todo plastificantes y retardantes de llama, y que son de los principales tóxicos asociados al plástico, que es lo que queremos ver. Aquí sobre todo lo que teníamos era una colonia en la que teníamos la oportunidad de relacionar la presencia de plástico en en el tubo digestivo en general, y es de lo que es parte del trabajo que estamos haciendo, y compararlo luego con la presencia de esos tóxicos, o sea, la cantidad de plástico que encontramos en los digestivos, relacionarlo con los análisis de isótopos estables para ver cómo de antropogénicos son ese origen de la dieta, y compararlo luego también con variables que tienen que ver con la presencia de esos tóxicos relacionados con el plástico en diferentes tejidos, ¿vale? Pero claro, lo ideal sería tener un montón de colonias de aves de muchos sitios, pero no es el caso. Ahora mismo quizás podamos tener acceso a muestras de la isla de Tarifa, que allí pues sí, lo que pasa es claro, individuos muertos podemos conseguir de muchos sitios, pero individuos que estén directamente eliminados de la colonia, porque claro, con los individuos muertos no podemos tener una idea realista, ¿no? de si la dieta que estamos identificando pues tiene que ver con pues bichos que están enfermos o algo así. En este caso lo que tenemos son bichos que se han matado directamente con un programa de descarte, y entonces es muy representativo lo que encontramos ahí. Son bichos que han podido estar pues recién casi comiendo en los sitios, ¿no? Pero claro.
Solo una puntualización, que yo creo que lo que tú estabas planteando, o sea, nosotros la manera de aproximarnos, aquí hay una limitación, que es en qué tejido vamos a medir esos contaminantes. Estos análisis los va a hacer el Tarrat, ¿no es el apellido? Sí. Entonces, ellos tienen puesto a punto la metodología para una serie de tejidos, no para cualquier tejido, sino para una serie de tejidos. No, en principio, lo que van a analizar en cuanto a las poblaciones, digamos, en ese gran muestreo que hemos hecho nosotros, donde hay un gradiente, donde estamos encontrando colonias, donde estamos encontrando más plástico en las egarópilas de las nidos y otras menos, la aproximación que tenemos nosotros para seleccionar como colonias más sanas, digamos, y otra es esa, no tenemos un lugar que nos sirva de referencia de contaminación cero, o sea, no lo tenemos. Entonces, en esas colonias nosotros estamos trabajando con animales, o sea, haciendo un muestreo de egarópilas, nidos y demás, y también muestreando animales que luego se le marca con GPS, se toman isótopos y demás. Y esos animales, los tejidos que les estamos tomando, donde ellos puedan medir contaminantes, son el aceite de la glándula oropigeal, básicamente.
Si yo estaba hablando solamente de esta parte, que es la parte de la necrosia, que es de lo que yo creo que tú te referías, ¿no, Pedro? De la parte de necrosia. Las necrosias solamente las podemos hacer con esta población porque es de la única que existe en datos. No tenemos bichos descartados de otros sitios.
Y con respecto a lo que tú decías de comparar con ejemplares de las colecciones, que claro, tiene una escala temporal a lo mejor de casi 100 años, sería estupendo, pero ¿con qué tejidos podemos nosotros? Plumas. Ahí podríamos trabajar con plumas, básicamente. O
parte blanda.
blandas, si en algunos casos pudiéramos
No,
mostrar.
esa era la pregunta, si es posible o si lo habéis pensado.
Tendríamos,
o
esto
la-
es un tema que podríamos, después de esto, quizás profundizar, si a través de la colaboración que tenemos con esta mujer, de intentar empezar a medir esos contaminantes derivados de los plásticos en otros tejidos, ¿no? Que además son análisis bastante caros y complejos. Pero bueno, a un futuro se puede plantear, ¿no? Porque es verdad que ahí hay un material que sería estupendo poder disponer de él. Pero no tenemos un sitio de contaminación, digamos, cero de referencia. Eso no lo tenemos ahora mismo.
Sí, bueno, well, I'll get back to English. So my question was not a criticism, but just thinking that probably it is impossible to find a place on Earth with zero contamination. So the point is to have the possibility to select some control where you can assess the background contamination that you have in, so to speak, natural conditions, and then how the situation of colonies that are quite close to the landfill that multiplies the contamination level. But it's just a question, not a criticism with the approach, of course. That is perfect.
Manuel, great. Thank you so much for the talk. I really like how you're trying to integrate all different aspects of it. So I think that's great. But I'm bounding on Pedro's issue. I think if you don't have a control, so I'm guessing experimentation is out of the question. There's no captive animals that you could experiment with, or in, like, Jerez, el Zoobotánico de Jerez, or
Cañada de los Pájaros. It's something that we have in Manuel, for example, for the movement data, and we also got analysis of the cattle egret, because some of them are marked in Costa Ballena, and they are moving normally, but we have 2 of them marked in Cañada de los Pájaros, where they are eating the cleanest way possible. So, and we also got sample from them. So it's something to compare, for example, with the cattle egret. It's something tricky talking about those animals that are always foraging in the, say, urban areas. But yeah, it's something for sure to keep in mind and to look for those control
because the
samples.
other approach would be if you have, if you're just relying on the necropsies, you would have a gradient of how much plastic an individual was showing, and what the parameters that you're measuring of the severity of the impact would be. So, but with 45 data points,
And only one colony.
right?
And species.
Well, but at least if the sampling is random, because if this takeout of the individuals is kind of random with respect to plastic, at least it's not like animals that appear dead, but you just, so it could be, And the other thing is, maybe if you'd have like repeated sampling of the same individuals, and you're taking both fecal pellets and some kind of plasma or some kind of repeated measures, estimate of the body condition of the animal, maybe you could see whether there are periods in which they have greater plastic deposition match with, I don't know. So because it's going to be associative in the end.
That's another point of, for example, sampling in breeding and wintering season to see if there is any difference between the way they are foraging during winter and during breeding, to have also something to compare with. But yeah, you're right with this.
Awesome.
Lots of chicks. It sounds like you're sampling lots of nests. You're watching them. Do a lot of the chicks die? Can you look at that?
Fortunately, this last year we only saw 2 chicks with legs amputated in the Salabajo ringing, for example. But yeah, a lot of issues are related with this. We also saw 2 of the chicks with malformation in the peaks, for example, for trying to eat something that they couldn't, like plastic related, because all the nests were full with plastics. But this last year, not also because of the rains, because there were more water this last year, so they were foraging a little bit more, maybe in a natural way. So the fathers were bringing, and they didn't need to go more to landfills. But yeah, the weaker the chicks are, more exposure they have to plastics, and if they are more ill, we can easily find dead chicks in the nests while they're ringing.
So do you look for plastics in the digestive tracts of those dead chicks?
No. No, and it's something that we can aim to do too.
Okay, so more questions? Okay, so I think that we can end now. So thank you, Manuel, for this talk, and thank you everybody for coming.